So, good afternoon and welcome all participants to Walayalak University International Webinar 2023. My name is Nichiwan Kromgel, Academic Officer of Center for International Affairs. So today the webinar is the third webinar in a series 1 of Walayalak University International Webinar 2023. Today we are going to be exploring the role of mitochondria in human being life and aging. So in follow-up webinar, we are going to be looking in more details of the evolutions of life on Earth. The scientists believe that the universe began 13.8 billion years ago, and Earth was born 4.6 billion years ago. Early traces of life were found soon or as later in 4.1 billion years ago. So do you believe at what the scientist says? Then we also discuss about the birth of mitochondria. Mitochondria are membrane-bound cell organelles that generates most of the chemical energy needed to power the cell biochemical reactions. So I'm glad to be joined by my colleague, Dr. Hideyuki Majima. He is from the School of Allied Health Science, and he is the speaker for today. So to kick the thing off, this session is going to last about 1 hour. So now may I introduce you to Dr. Hideyuki Majima. He graduated PhD in oncology from Nikon University, Japan, and now he is the lecturer at the School of Allied Health Science. Now I would like to turn the stage over to Dr. Hideyuki Majima and ask him to start the webinar. Thank you.
Thank you. Thank you for introductions. So anyway, I'm going to talk about the roles of mitochondria in human beings' life and aging. Oh my goodness. I can't move. Okay, this is abstract. First of all, I'm going to start from history of Earth. This is a 4.6 billion years. Then you can see it. So now, and then you can find this is the first life was born. It's for sure the first life was born on Earth because we are here. So then, so this is the next one. As you can see, the same scale of 44.6 billion years, but this is concentrations of oxygens about like 2.5 billion years ago, and increased. It's keeping like a 20.5%. So this is a combined pictures of the first slide and then second slide. And then scale is the same as 4.6 billion years. Okay, and then human being just was born, but before oxygen concentrations increase. And then you can take a look, and then here, and then mitochondrion was born. Okay, so when the first life was born on Earth, there was no oxygen at all. As people may think we need oxygen, but later the oxygen's percentage increased. Then we started to use oxygen as a source of energy using mitochondria. So, okay, And then, and one more point, the oxygen is a harmful molecules. Like I say, ions, ion lost by oxygen. It's harmful. So that life on Earth established the protections against oxygens. And then finally, they used oxygen for the source of energy. Mitochondria, like us was born 1.4 billion years ago, bacteria getting inside the cells is archaea, okay? And then establish mitochondria, finally. And then like before the born of mitochondria, we produce only 2 ATP by glucose, from glucose, only 2 ATPs. And after established mitochondria, we can produce 2 plus 29 ATP using mitochondria. So cell can produce more ATP energies. Then that's why that could make like a animal plant and a multiple cells life. So this is mitochondria, and we have electron transport chain in mitochondria produce more ATP here. The inner membrane of mitochondria. It's inner membranes, outer membrane and inner membranes. Okay. Then, like looking at mitochondria, it's the producing ATP is not only one functions, a lot of functions, fatty acid, ligations, phospholipid synthesis, so electron transfer, this is the produce energies. And then pyruvate dehydrogenase complex, so many functions in mitochondria. It's amazing. Mitochondria has DNAs. The people know it's we have DNA in cells, and then nuclear. Then this one is, but besides that DNA in nucleus, we have DNA in mitochondria. Because this is used to be one different life. So still has DNA inside. This is electron transport chain. And then like actually the mitochondria can produce the 13 proteins. And this is electron transport chain to produce ATP, the machines. But yellow part is made of mitochondria here. 13, there is 13 genes produce 13 proteins. It's like here. Other blue one is come from nucleus. This is inner membranes, and here is outer membranes. Actually, this electron transport chain, electron move, and finally produce ADP plus ATP, okay? And then another thing, this is not complete machine, and then the 2 to 3% of electron leaks, and then produce superoxide here. This is inner membranes, and here is outer membranes, actually. And this is intermembrane space, so-called. So then the superoxide is produced here. And superoxide, because 2 to 3% of electron leaks from electron transport chain, and the oxygen, and that's bind to the electron, then produce superoxide here. And then superoxide produce more reactive oxygen species, ROS. So this machine can produce more ATP, but the producing ATP, this one produce also reactive oxygen species. That is problem. Okay. That's why that people has aging. Reactive oxygen species, or the different words, free radicals, and they're almost the same meaning. Okay, and then but when you say reactive oxygen species, that's everyone is radicals has O. But if you say free radicals, that's maybe you don't necessarily have oxygen, but also that nitrogen also. ROS damage protein is as protein oxidations, repeat oxidations, and then DNA damage. Then finally cause aging, cell death, calcinogenesis, and then neuronal degeneration. Okay, mitochondria, very important organelles to produce ATP, but at the same time that cause aging, cancers, neuronal degenerative disease, diabetes, infertilities, rheumatoid arthritis, Crohn's diseases, atherosclerosis, and cataract, many diseases. All these people know diseases, but long time people try to find bacteria. Bacteria cause disease, but no one could find any bacteria. This one was oxidative generative diseases. Okay. So mitochondria cause superoxide, and mitochondria is a major source and agent superoxide radicals. And we talk about new superoxide theory, because as we do science has history and discover superoxide dismutase. This is a very famous Dr. Friedrich is still alive in the US. It was 1969, and then manganese SOD was discovered. Same person, same as famous Dr. Friedrich, and then also superoxide is not strong oxidant. They found out this 1981, and also NO was found as 1987. Then that's the founder of 3 scientists is received Nobel Prizes. And then peroxynitrate, also that's potent oxidants that's found in 1990. Then manganese SOD protects against cell death. This is my paper published in 1998. Nobel Prize. The 3 people received Nobel Prize in 1998. This is my paper published, the first papers published in 1998. That's that ROS was generated from mitochondria and cause apoptosis for the oldest papers in the world. And also some other papers by us, mitochondria produce ROS, but if you have, like I said, damage in mitochondria, so as you produce more ROS. Aging in the mitochondria, SOD. So, and then I'm going to show you, like I see, this is that's enzyme here, and then this is SOD, superoxide dismutase, and this is lifespan, aging, and you can see this is activity. SOD is increasing. It looks like as the SOD is related to aging lifespan. The second one, superoxide dismutase, correlation with lifespan. It's like this. This is SOD. Is this depending on age, that you can see increase of SODs. Okay, that's the aging and then superoxide dismutase, again correlated. This is our papers, the review papers of our mitochondrial superoxide theory, but oxidative stress diseases and aging, published in 2015. Threshold hypothesis. This is this is age, okay? And then the function decrease is as a function of aging. And if you have any kind of damage, it's a mitochondrial disease patient, it starts earlier. And then this is damaged mitochondrial DNA is as function of age. Okay. Mitochondrial DNA copy numbers. So it's various point, but it looks like as a stable. So mitochondrial DNA copy numbers it's difficult to say. This is dependent on the age. This one's the same. This one the same. So morphologies. This is young and old, just mitochondria. Obviously, old one is getting damaged. Okay. Mutant mitochondrial DNA. Yes, because it's a DNA, it can get mutations. So this is a human, this is mouse, and then mutation. Fractions mutated mitochondrial DNA %. So this is looking at the mouse is start earlier because they don't live longer. Human beings live longer, as you can see. Damaged mitochondrial DNA increase as a function of age. This is another DNA damage. Also, you can see increased damage. This is liver, kidney, and intestine. Okay, that's damage increase. This is a complex 4 deficiency. Complex 4 get damaged. This is among electron transport chain. That's increased, dependent on the age. And another mutation for mitochondrial DNA, this is specific mutations. Look at increase. So if you get age, you get increase of mitochondrial DNA mutations. Okay, this is a final slide, actually, conclusions, aging in the mitochondria. DNA copy number, it is stable. This is, but other morphology of mitochondria and the fraction mutant mitochondrial DNA, mitochondrial complex 4 deficiency, and then proportions of 4977 mitochondrial DNA mutations also increase. This is conclusions. This is a picture again for the old mitochondria. Okay, finished. Thank you very much.
So thank you. Can you like there share the picture, share the slide like one by one, or?
No,
No.
please not, because I'm going to write papers.
Oh, okay. So I think we have one guest. Do you have any question to ask during this? I don't know his name. Okay. So all right. So thank you very much for your information of the mitochondrions, and I will, after this, I will upload it on YouTube channel of the CIA. So maybe the guests can rewatch it later, and I will send the links and information to our friends, our colleagues from to our partner university as well. So all right. So thank you everyone for joining the webinar today. Thank you. See you next time. Thank you.
Thank you.